Oh, thank you so much, Sue. It's a very generous introduction. So it's a huge pleasure for me to kick off this first session of the day, introduce our first invited speaker. Dr. Mariana Silva Artur is the leader of the Seed Resilience Group within the Laboratory of Plant Physiology at the Wageningen Seed Science Center, at Wageningen University in the Netherlands. I first met Mariana back at DesWorks in 2024, which is a desiccation tolerance workshop in South Africa. And Mariana was one of the first people to welcome me into this community, and for which I'm so grateful. Mariana has done fantastic work studying the roles of Leia proteins, both in resurrection plants and orthodox seeds. This work has included the bioinformatic exploration of a xerophyta genome, the mapping of Leia protein gene families across plants, and more recently, her group has developed a transcriptomic atlas of seed maturation. Overall, Mariana and her group have been at the forefront of studying how desiccation tolerance regulation evolved across the plant kingdom, all while building out fantastic tools and resources for our community. Today, she'll share some of her recent work using seed maturation as a system to study resilience and evolution. So with that, I'll let Mariana take it from here. Thank you.
Thank you very much, Olivia, for the nice introduction. Can you all hear me?
Yes.
Check. Thank you. Thank you very much. I will share my screen. Let's see, share and share. Yeah. Do you see my presentation?
Beautiful.
Okay. Yes, thank you very much, Olivia, for the kind words. Really happy to hear how the DesWorks meeting from last year resonate. And I also would like to invite to thank everybody for being present here, and also the organizers for the invitation to present my work and the work of my group in this first WALII Symposium. That's a very nice opportunity to present here our work. So I'm an assistant professor here at the Laboratory of Plant Physiology, as Olivia mentioned, and my group is starting to explore a bit more seeds as our system to understand desiccation tolerance. So today my talk is titled Drying to Survive, Seed Maturation as a Unique System to Study Resilience and Evolution. So here I also would like to show you some of our recent works and some directions that we are going on this research. So as you probably have noticed, I find seeds a very fascinating biological system. And I like to see seeds in the center of the plant life cycle, because from a seed, there is an embryo, which contains all the information necessary for a plant to grow and continue its life cycle. What is also quite interesting, an interesting aspect of seeds that fascinates me is their ability to tolerate desiccation. And in my vision, this desiccation tolerance ability of seeds works as a pause in this plant life cycle, which might enable plants to reset some of their programs to be able to survive in the environment. So seeing seeds with this context of importance, we can also be aware that seeds are important for biodiversity conservation, and they form the basis of plant survival and adaptation in different environments, but also for our agriculture, economy, and food security. So a question that I have been studying during my PhD, and that now my group is investigating as well, is a very basic fundamental question, which is, when do seeds become desiccation tolerant? So to address this question, I would like to introduce to you seed maturation and seed development, especially for the people who are not familiarized with seeds in the audience. So seed development is usually characterized or divided into two major phases. The first phase is the embryogenesis, or also called morphogenesis, when there is a formation of an embryo, cell divisions, and activation of the zygotic genome. And after embryogenesis, seeds enter to this maturation phase of the development, where there will be events that leads to the accumulation of nutrient reserves, which is also called seed filling at the beginning of the maturation. And also later, some species will undergo a drying after the maturation or the seed filling, which is called desiccation phase. And all these events will take place whilst the seeds are still in the fruits in the mother plant. So during seed maturation also, a couple of very interesting traits, which we call seed survival traits or seed quality traits, are acquired. And these traits are very relevant for seeds to prepare them to survive for long periods of time, to become this point of resetting of the plant life cycle that I like to do this comparison, but also to allow them to remain in the soil dormant for the species that produce the seeds that remain in the soil. And also for their resilience to biotic and abiotic stresses, for their germination, and for them to survive in dry environments, which is the desiccation tolerance ability of seeds, which I think we are all here familiar with. So what this shows to us is that seed maturation is a very dynamic phase, but also very active physiologically and molecularly. And in our group, in the Wageningen Seed Science Center, we have studied seed maturation in relation to these different traits, which Olivia mentioned, where we identified different regulatory programs which coordinate the acquisition of these different traits during seed maturation using Arabidopsis thaliana as a model. And this work is currently in bioRxiv, where we generated some resources, including RNA sequencing from multiple genotypes, which are important for the control of the acquisition of these traits. But today I want to discuss and present a little bit of work that we are developing in my group currently, where we want to investigate what is the evolutionary basis for this ability of maturation, or this characteristic of seed maturation to be such a dynamic phase physiologically and molecularly. So to address this point, I would like to introduce to you phylotranscriptomics for those who are not familiar with this approach. Phylotranscriptomics, nothing more is than a combination of phylogenetics and transcriptomic analysis, and it allows us to infer evolution through gene expression patterns. So I brought here a model plant, Arabidopsis thaliana, which is our model plant for research in plants and also in seeds. And let's suppose Arabidopsis thaliana has 4 genes, very simplistic, gene D, C, B, and A. Gene D is present only in Arabidopsis, so it's a very species-specific gene. Gene C, for example, is shared across Arabidopsis and other dicots and monocots, so angiosperms. So, for example, here's Rhophytes schlechteri, which for the people working on resurrection plants, that might ring a certain bell. Gene B dates back to the ferns, and gene A dates back to the ancestral of land plants, for example, an aquatic greenhouse gene. So from this information on the origin of these genes, we can build a phylogenetic relationship, and in this case, it's a phylostratigraphy profile. And in that sense, we assign phylogenetic ranks to these genes. So gene A is a more ancestral, so it gains this phylogenetic rank called phylostrata, a bit lower, so it's not phylostrata 1. And we go along with the origin of these genes. So gene B belongs to phylostrata 2, gene C phylostrata 3, and the most recent or younger gene in this phylogenetic scale belongs to phylostrata 4. So as I said, it's possible then to create this relationship between these genes that we find the origin, we identify their origin, and then we can overlay or we can combine this information with gene expression. And this allows us to then perform this phylotranscriptomics analysis by calculating what we call a transcriptome age index, which nothing more is than the weighted mean of the phylogenetic rank of the genes by their expression pattern, so by their expression level. So this allows us then to identify the origin of genes present in a certain transcriptome. So here I brought an example as well. I like to do these examples to help it on the visualization of this. So considering this phylostratigraphy, genes which are older, they will receive a lower transcriptome age index. And genes which are more young or younger genes, they will receive a higher transcriptome age index. And then if we perform transcriptomics analysis, for example, RNA sequencing in an ontogenetic profile, so for example, leaf development, so we have a younger leaf towards an older leaf, and we identify this gene. So for example, in the first sample of RNA sequencing, this younger leaf has an enrichment of genes which are expressed that belongs to the older phylostrata, so the phylostrata 1, so they receive a lower transcriptome age index, and this can follow throughout this phylogenetic, this developmental time course. And for example, this older leaf has again an enrichment of older genes, so it will receive a lower transcriptome age index. And we can look at these different profiles to identify any evolutionary signals in our transcriptome. And this is possible thanks to the availability of this Mighty AI, which is an R package developed by the group of George Drost, which has been very handy for us to understand evolution through transcriptomic data information. So this phylotranscriptomics analysis has been used in animals and all different types of foods, and also in plants, in different organs, and in seeds, which is my prior model system to study. And it has already been reported in the literature that seeds show what we call the molecular hourglass pattern in their development, in the embryogenesis and in the germination. And this hourglass pattern is a reference to the morphological hourglass pattern, which has been described for animal development, which states that during the development of multiple different animals, they converge into a similar morphological state, which is called a phylotypic state. And this is supported by a conserved transcriptome at this state. So in our group, we found this very interesting. And because we have generated the seed maturation transcriptome with high resolution of Arabidopsis thaliana during seed maturation, we asked this question: is it possible to use phylotranscriptomics to reveal these evolutionary signals during seed maturation and across this entire seed life cycle that we named, which is going from embryo to the germination of seeds? And I'm very happy to answer this question by saying yes, phylotranscriptomics can reveal very interesting evolutionary signals. And this is a work that has been published recently in Plant Cell, 6 days ago, actually. And I'm really happy to share this and highlight here our brilliant candidate, PhD candidate, Asif Ahmed Sami, which did a great work in putting this work together and developing also these ideas further. And I would like also to invite you to watch his presentation later on during the day. So in my presentation, I want to show a little bit of our findings and how this links to the regulation of desiccation tolerance in seeds. So I would like to show some of our major findings. So we performed this analysis using the available data for embryogenesis and germination for Arabidopsis thaliana. So we repeated these analyses that have been described in the literature using our own RNA sequencing pipelines. And what we could see was that indeed this hourglass pattern was statistically significant for both phases. And this was done using this TAI package, where it allows us to statistically test for specific profiles in the transcriptome. And what was interesting for us was to see that when using our seed maturation data, we could not see this hourglass pattern of development. Actually, it was not really significantly different than a flat line. So it was similar to a flat line, actually. So it was not an hourglass pattern. But what was interesting for us was that when we combined this data from these 3 different phases to perform these statistical tests, we could see a significant reverse hourglass pattern of this development of the seed life cycle, showing that seed maturation seems to have higher enrichment of genes from younger age expressed in comparison to embryogenesis and germination. And we also investigated what were these genes, and also in their genetic sequence, analyzing their synonymous and non-synonymous substitution rates, where we could see that also during seed maturation, the enrichment of these genes were also for genes which were more rapidly evolving. So next, we asked how conserved was the expression of these young genes during seed maturation. And for these, we used available data sets for embryogenesis, maturation, and germination for rapeseed and tomato, and also for maize, where we could see that indeed this reverse hourglass pattern was statistically significant and conserved across dicots and monocots. And we also later identified this pattern in data sets that have been published for barley and soybean, so RNA sequencing data sets for seed development and seed maturation, where we also found this conservation of young genes during seed maturation in multiple plant species. So one part of our work was also to try to understand what kind of tissues drive this reverse hourglass pattern of the plant life cycle, and also this high enrichment of younger genes during seed maturation. And thanks to the availability of RNA sequencing data for tomato, maize, and barley of tissue-specific RNA sequencing during seed maturation, we could observe, was quite interesting, that it was also conserved the fact that the endosperm was the major contributor of this high transcriptome age index in multiple species, showing that these younger genes seems to be preferentially expressed in the endosperm. But of course, we need more data sets of more species to really investigate also the impact of these younger genes in the endosperm, and also to try to infer a little bit about the role of domestication on selecting seed traits and especially selecting characteristics of the endosperm. So a question that we also wanted to address, to drive into a little bit more the mechanism behind this reverse hourglass pattern, was to understand what drives the reverse hourglass. And the approach we took was to sort the genes that were highly expressed during seed maturation and start removing the top highly expressed genes. And what we could see was that by removing only 5% of these top highly expressed genes during seed maturation was sufficient to abolish this reverse hourglass pattern, meaning that these 5% top highly expressed genes were responsible for this reverse hourglass pattern during seed maturation, and also in relation to embryogenesis and germination. So we delve a bit further to see what were these genes, what were they doing. And here we reorganized them a little bit into different classes. So we have younger, intermediate, and older genes. So we reclassify them based on their phylogeny or their phylogenetic rank. And summarizing this image, this figure, what we see is that for the genes which were older, which were amongst the top highly expressed genes during seed maturation, but they had an older origin, we could see mostly biological processes which are more conserved or ancestral. So for example, cytoplasmic translation, response to hydrogen peroxide, and embryo development, which are in the plant phylogeny more ancestral, let's say, in relation to younger biological processes, which characterize more species-specific traits and adaptations. So here, the two groups of biological processes that we identified as younger or driven by younger genes were, for example, mucilage biosynthesis and seed coat development, which are also very characteristic of the species. And another very interesting finding was the identification of some biological processes that seems to have multiple origins. So they are in seed maturation, they encompasses old and younger genes. And those processes seem to be involved with the combination of stress and development. So two here that I highlight, which were also highly enriched, are response to abscisic acid and response to water deprivation. So this was very interesting for us because as we are interested in desiccation tolerance, this led us to think that most likely these seed survival traits, such as desiccation tolerance, might be coordinated by a combination of older and younger genes, which allows them to be shaped during the development of the seeds. So we wanted to also further understand how this combination of this evolutionary analysis could give us some physiological insights and also more molecular insights. And from the literature, we knew that has been already described already in 1994 that the regulation of desiccation tolerance is coordinated by two pathways in synergy, which is through the hormone abscisic acid, which is one of the processes that we identified in our phylotranscriptomics analysis. And this maternal or applied abscisic acid leads to the expression of abscisic acid responsive genes during maturation and protein synthesis, in combination with the maturation itself or the slow drying that happens at the end of the maturation. And in combination, these two pathways will lead to what we call the desiccation tolerance. So there are two regulatory programs coordinating the phenotypic appearance of this trait, which is desiccation tolerance. So for us, this was very interesting because it seems that in seeds, this desiccation tolerance has multiple origins. So we are now investigating this further at the molecular level, but also at the physiological level. And our approach was to perform some physiological assays on maturing seeds and submitting them to different drying treatments. So we have seeds submitted to fast drying, to slow drying, to see what were their physiological responses to these drying treatments. So here, I want to show the first part of this developmental program that coordinates the desiccation tolerance, which is in a wild-type plant, where we see that in fresh seeds, they are capable to germinate, or they acquire their germination ability already at 12 days after pollination. But if we submit the seeds to a fast drying treatment, they are not capable to survive fully, the seed population, only after 16 days after pollination. But if we perform a slow drying treatment prior to this fast drying treatment, we can see that already at 12 days after pollination, it's possible to induce this desiccation tolerance in premature seeds. So showing that slow drying accelerates desiccation tolerance acquisition. And we also perform these assays using a mutant that is deficient in the production of abscisic acid. And what we could see is that the mutant, when we perform a fast drying treatment, the mutant also shows at 14 days after pollination lower survival in response to fast drying. But when we dry this mutant using this slow drying treatment, we could see that there was a partial rescue of the desiccation tolerance of this population, or a partial increase in the desiccation tolerance of this seed population, showing that desiccation tolerance can be partially induced by this slow drying in an ABA-independent manner. And this is work that we are performing now to try to understand what is this ABA-independent pathway that regulates the acquisition of desiccation tolerance during seed maturation. But what these findings shows to us is that drying seems to be a major force driving desiccation tolerance acquisition during seed maturation. And of course, linking this back to our phylotranscriptomics profile, the question is what genes and how old are these genes and what are these downstream pathways is a cliffhanger. I won't tell you that yet because it's something we are finalizing now. And I hope to be able to show you a little bit more. But together, we have shown that it is possible to combine phylogenetic information with gene expression data to identify regulators of desiccation tolerance in seeds, and also to study the evolution of this trait in plants. So in summary, I hope I could show you how we are using seed maturation as our system to study evolution, evolutionary innovations, and also desiccation tolerance and resilience overall of seeds to multiple stresses. And that we think we have put the seed maturation in the spotlight again for the studies of desiccation tolerance, because we could identify that thanks to the seed maturation expressed genes, we can see this reverse hourglass pattern. And we propose this out of the seed hypothesis, where it seems seed maturation acts as this evolutionary landscape to test younger genes and facilitate functional specialization in plants and in seeds, and also likely allows seeds to express this desiccation tolerance phenotype, which is also extremely important for crop domestication and also for plant adaptations. And together, we are now investigating how drying might act as this evolutionary force coordinating the expression of genes which are required for desiccation tolerance acquisition during seed maturation in plants. So with that, I come to the end of my presentation. So I would like to thank you for your attention. Thanks again, all the organizers, for the invitation. Special thanks to my colleagues Asif Ahmed, Sami, Leo Willems, and Leonie Ben Sink, and all the members of the Wageningen Seed Science Center and the Laboratory of Plant Physiology for their amazing work environment, and also our funding agencies. So thank you very much for the opportunity.
Thank you so much, Mariana. That was a beautiful
Yeah.
talk. I really loved learning about the phylotranscriptomics
Thank
and
you.
seeing your paper, seeing aspects of your paper here in the presentation.
Thank you.
Um, I have, it looks like Kent and Aaron kind of beat me to one of my questions. So I'll start with Kent's
Mhm.
question. What exactly was the slow drying treatment you used? How slow and how dry?
Yeah, yeah. So I didn't mention this here on purpose because we have done other several drying treatments, but now I can disclose that because we are in the process of writing this manuscript. So our slow drying treatment consists of maintaining the seeds in the fruit before
Yeah.
removing them from the fruit for this fast drying treatment. And in our work, we have quantified abscisic acid in the tissues of the fruits and in the seeds to also understand the relationship and what happens during the drying, during the maturation. So this was a tool that helped us to assess this low drying treatment because by removing the seeds, our Rhabdopsis seeds are very small and they dry very fast. So we have tested this in different ways and we would never manage to slow dry them without maintaining them in the fruit. But this way we can also assess the effect of the maternal tissue in the
Mhm.
acquisition of desiccation tolerance, which is part of the
Mhm.
hypothesis of desiccation regulation. Yeah.
Yeah. And how long would that be, that duration in the fruit?
Yeah, yeah. In total, it is 4 days. So it's
4 days.
2 days, 2 days slow drying, 2 days fast drying. Yeah.
Okay. Fantastic. And an anonymous attendee asked which ABA mutant was used in the slow slash fast drying assay.
Yeah, it is an ABA deficient mutant that I can't disclose at the moment. Sorry for that. But it is a mutant that produces lower level of abscisic acid. So it's a deficient mutant, is not a null ABA mutant
Mm,
because a seed mutant without ABA can't develop. So it is a mutant that produces ABA, but very lower level during maturations, also specifically compared to the wild type plant. Yeah.
fantastic. And Cesar wrote, Hi Mariana, wonderful presentation. I wonder which genes were induced after slow drying in the ABA deficient mutant that tolerated desiccation when
Yeah.
subjected to slow drying.
Yeah, yeah, that's something we are
Hmm.
identifying now. We are investigating it now. We want to combine this actually with our phylotranscriptomics analysis because our hypothesis is that we will identify maybe some phylogenetic, some evolutionary profile or pattern in the activation of these genes. But we haven't yet. We are working at the moment, but we haven't yet validated these genes to prove that they are indeed part of this ABA-independent pathway. Yeah.
Okay. And Cesar's final question is, did you find layer proteins induced? And if so, which groups?
Yeah, yeah. We have identified LIAs. We also identified LIAs in the slow drying treatment. Yeah, at the moment it were a combination of classes, so I can't really mention which ones specifically. But that's something we can look closer as well. Thanks for the question.
Hmm. And I see, so I think there's one question in the webinar chat that I'll get to, but the last question in the Q&A
Mhm.
from Mel, have you looked for some of these genes in non-angiosperms? Great question.
Yeah, that's a really good question. So I think you mean the genes from the phylotranscriptomic profile, I wonder.
I presume
Uh,
so. It
yeah,
wasn't
yeah.
specified.
Yeah, yeah, we did what we found by looking at well, we looked only in the monocots and dicots species. Asif will present later on, so here I already leave you with waiting until the later presentation where we have performed the phylotranscriptomics profile on resurrection plants. So we did characterize some of these genes which are found in seeds also in resurrection plants and some of these biological processes. Yeah.
Oh, fantastic. I can't wait to see it.
Yeah.
Um, and then Prakash asked in the webinar chat how how high seeds can dry until they hold tolerance and preserve germination capacity. Or rather, how dry they can get. Yeah.
Yeah, they can get really dry. I think from our general definition of desiccation tolerance, for example, Rhabdopsis seeds, they dry up to down to 8% of their initial moisture content. So it is very dry and very low moisture content of the seeds. But this is possible throughout the maturation. So this is a bit of a cumulative characteristic of the seed population. So as I showed, around 16 days after pollination for Rhabdopsis thaliana, those seeds can fully dry, what we call full dry, which is down to 8%. But our slow drying treatment seems to enhance the possibility of the seeds to retain their germination capacity earlier in the maturation.
Fantastic. Thank you so much, Mariana.
Thank you. Thank you for the questions. Yeah.
Thank you. With that, I don't see any more questions, and so I'll transition to our first contributing talk or contributed talk.